Welcome to Applied Photophysics webinar. My name is Cam Hick, Sales and Marketing Support for the U.S. Today's webinar is Speed and Precision Fast Kinetic Studies for the SX Stop Flow Spectrometer. Our presenter today is Dr. Lindsay Cole, Research Director for Applied Photophysics. If you have any questions during the webinar, please ask them to the question box on the right-hand side. Lindsay will answer as many questions as possible at the end of the webinar. We plan to keep the webinar to about 45 minutes. All questions that have not been answered will be answered directly to you through email. Please direct any questions after the webinar to info@photophysics.com. This webinar is also being recorded, and a copy will be sent to you through email in a couple of days. Also, it'll also be on our YouTube channel, posted there for your viewing. Once again, thank you for attending, and I will turn this over to Lindsay. Lindsay?
Thank you, Cam. Yes, I'm Lindsay Cole, the Research Director at Applied Photophysics, and I'm here today to talk to you about the SX20 use in fast kinetic studies. So first off, who are we, Applied Photophysics, and how do we come to be doing stop flow? Well, Applied Photophysics was originally founded from the Royal Institution of Great Britain by Lord George Porter, who actually got a 1967 Nobel Prize in Chemistry for his work in fast reaction kinetics, in particular laser fast photolysis and light-induced photolysis, and then looking at the fast reactions. But this long history in fast kinetics eventually led Applied Photophysics to develop other fast kinetic techniques, in particular the one I'm going to be talking about, of course, stop flow spectrometers, and in particular the latest version of that in the long line of stop flow systems from Applied Photophysics, the SX20. Now, stop flow is still a widely used technique in a lot of different areas of chemistry and biochemistry and biophysics, and still an important part of a number of high impact publications and research programs worldwide. And just here's some very recent high impact journal articles using an SX20. Right, so my talk today is going to have a first part which will actually talk about the product and the SX20 itself and what stop flow is at a very simple starting level, and then we're going to have a look at some case studies of some simple and more sophisticated techniques of stop flow, looking at some simple absorbance chemical kinetics, going into a more sophisticated examples of fret-based techniques in fluorescence, and then a very interesting utilization of fluorescence polarization, all these different modes of optical probe are available on the SX20 and are very good. Right, what is stop flow and where is it used? So what is it? It's to study fast reactions in solution. The timescales, milliseconds, sort of 1 millisecond to several minutes is the timescales that we're able to use. And on this chart here is looking at different kinds of approaches that you can use to study chemical kinetics, essentially reaction rates and so forth, from different techniques, from pulse lasers through to classical methods, being simply mixing things together and observing the change in a spectrophotometer, for instance, to flow methods, which is effectively what the stop flow is. And it's in the range of effectively that milliseconds to minute time scale. At its simplest, two reagents are mixed together in a flow, and then the flow of those two reagents is stopped, and the reaction is then observed from that stop in an observation cell, usually a fused silica cell. And it's normally optical signals that are recorded, and the change in those optical signals in real time is what is recorded, and then what is analyzed to give reaction rates and other things that can then be used to understand the chemical process under study. And the optical signals that we use are fluorescence, absorbance, can use CD in some some instruments, and fluorescence polarization. And all of these different techniques are available from APL. So, connected analysis of results can be determined by reaction rates, one or more steps. They can provide information on the complexity of the reaction mechanism. So effectively, how many steps, how many intermediates are on my reaction path? Is it A to B or is it A to B to C? So we have an example here on the right of a protein refolding experiment. This is where we take unfolded protein, dilute in a denaturing, dilute it down, and that induces folding of the protein. And in this particular case, it is actually at least two phases, a fast phase and a slower phase with two rate constants widely separated. This provides very good evidence that there is more than one process happening. There's an intermediate state and then going to the finally folded state. And you basically fit exponential curves to that, or a model that provides you and verifies that understanding of the process. There is also other things you can do with the rate constants, include looking at the energetics of what's happening and give some idea of, you know, the driving force of the reaction as well. But the top flow applications are fairly widespread in chemistry and biochemistry, but you know, and there's more than that I've listed here. But chemical kinetics generally, any kind of chemical process reaction that you want to monitor and see how its reaction occurs and what the intermediates are. That's an obvious one. Protein folding is another area that has a lot of stop flow traditionally and still is about understanding how proteins fold, what is the mechanism, and for that matter, how proteins unfold, and how that sequence and other aspects of the protein affect that trajectory of the protein folding process. Protein-protein interactions and actually ligand binding generally is a big area of stop flow research and understanding that process. And I'll come to an example later that will sort of highlight why stop flow is quite useful in understanding that process. And I think another area that I'm particularly familiar with is enzyme kinetics, understanding what's happening in an enzyme. And it's generally in that state, it's called pre-steady state versus what traditionally is called steady state enzyme kinetics. Steady state being your Michaelis-Menten kinetics. Pre-steady state is looking at the reaction cycle, but before it reaches steady state. And that you have to do faster than a second. Therefore, you have to use stop flow for that process, for that kind of application. So yeah, what are the applied photophysics systems? So this is a schematic of the SX20 stop flow design. Over here, we've got the two drive syringes I mentioned before. Effectively, we generally turn to syringe 1 and 2, or A and B. You have one reagent here, a second reagent here. And then what happens is this drive ram pushes the two together through this mixer. And as I said, it's in flow, and it'll actually go all the way through, pushing down that stop syringe. The stop happens when it reaches this hard stop. It can't move anymore. This triggers an electrical trigger that triggers the system to start acquiring data. So we know where P0 is, and we just look through this optical cell, which has now been freshly filled with just mixed reagents at about a millisecond. And that millisecond, or one so forth, is the dead time of the instrument. So that is the core of a stop flow. The optical measurements then can happen in this direction, which is a standard absorbance detection, or alternatively, at right angle fluorescence, right angle we can do fluorescence through another detector up here. And I'm going to show you examples of both kind of both data, examples of both later on. So that's the fundamental of it. Over there is the actual unit itself that actually has all these features in it. So you can see the syringes here, and there's actually the detection cell and so forth is the rear of the instrument over here, and here's the stop syringe. What are the fundamental advantage of the SX20 is it is quite a versatile design, and that is to do with how the cells are arranged. It has very high performance, and again, this is to do with the cell design and also the design of the whole system head itself and associated components. And it is a very reliable, robust system. And again, that's coming down to many of the aspects of design, including the whole all stainless steel design that is very tolerant of a lot of different chemistry. Right. So we have two core variants of the SX20. So on the left is, I guess, the standard system, the most common system used out there, and it's a xenon light source, very high intensity light source for all absorbance and fluorescence applications. It's a wide range source, and we usually coupling that with either a monochromator that we can select a particular wavelength for doing a particular absorbance or fluorescence experiment, or we can even do spectra. But this gives us great flexibility to pick any particular wavelength that will suit any chromophore or fluorophore that you want to observe. There's also an option to put on a photodiode array system that can acquire absorbance spectra at under a millisecond through the whole reaction course, so we can get the full spectra in absorbance mode. On the right there is a newer variant of the SX20 utilizing LED light sources. Now, there's a couple of advantages to this. They've got a high sensitivity simply because they tend to be very high intensity in the wavelength of interest. They are very stable simply because they have a rapid startup time because they're LED solid state devices, and that is an advantage. And it saves space because it's generally smaller. You don't need a large lamp, its power supply, and a monochromator. The disadvantage is, of course, you are losing the flexibility of a monochromator, which can go to any wavelength within this rather wide range, from effectively the UV to just near the near IR, red end. So let's actually get into what kind of data you can get out of it, what kind of experiments you can do. And here's a very simple, this is actually very simple absorbance chemistry we use that does show off a lot of what kind of data you can potentially get out of an SX experiment and what that tells you about the reaction and what you can actually get out of it. So this is actually a very simple experiment. It's actually a triethylamine nickel 2 complex over here. We mix it against acid, so that's in one syringe is the nickel, mix it against acid, and it will start to hydrolyze in actually 3 steps. And there's actually 2 intermediates, now starting material and a final product over here. And actually we can measure all of this in absorbance. And we can actually use the photodiode array, which is basically able to take spectra at sub-millisecond timescales. And so we get the whole spectrum in one. The other thing about it is our latest PDA is directly coupled to the instrument, providing better signal to noise overall. And that's simply because it is as close as possible to the cell and the actual chemistry that is happening. And if you do this, you get this rather nice but quite complicated spectral change over here, which is basically different time points and different intermediates going from A to B to C to D in this case. And actually, if you fit the data, you can get both kinetics, which is going to be on the next page, but also the spectra of the intermediates. Now, the point of this is that obviously we probably quite easily able to tell the initial intermediate and the final intermediate, but you can also get the spectra of the two starting material and the product, but also the 2 intermediates in between by fitting the data and effectively extracting what the spectrum of each intermediate is. And you can see we basically go progressively towards the red as the reaction proceeds in each intermediate becomes dominant. If you're able to understand what that spectra is telling you through separate experiments, then you can start to unpick what the intermediate is chemically, and that can give you a greater understanding of what your actual mechanism is, particularly of the intermediates. And if you can do that, that will be very insightful for a lot of different reactions and a lot of different processes. Understanding what those intermediates is can give great insights into what really is happening in your process. So here is the kinetic aspect of it. Now, this is not raw data. This is actually after fitting. And what is it actually showing you is the reaction coordinate of each intermediate. So in red, we're quickly depleting the triethylamine nickel 2 to the first intermediate that then goes to the third intermediate in green. And then at a longer time scale, so this is up to 2 seconds, at 42 seconds, we finally depleting the green intermediate and eventually going to the final product, at about 42 seconds in this light blue. And we've got 3 different processes happening, K1, K2, and K3, and this is the fitted rate constants showing the full process. So now I'm going to go into something a little bit more sophisticated. So fluorescence resonance energy transfer, or FRET, is a very useful way of approaching fluorescence experiments. The fluorescence excitation of a donor fluorophore is transferred to acceptor if they're less than 10 nanometers from each other. So if you're able to put 2 related fluorophores together with a different excitation and emission spectra, but the emission of the first one, the donor, overlaps with the excitation of the acceptor, you can actually use them as a molecular rulers to understand when they're close together and when they're far away. To facilitate this, and facilitate this in the SX20, we can supply the unit with 2 detectors. One looking at the fluorescence of the donor, the fluorescence emission of the donor, and the other looking specifically at the fluorescence emission of the acceptor. And this is the average here. There's one detector here and one detector here around the observation cell in here. And if we have bandpass filters that select specifically for the acceptor of the donor, we can actually observe whether this is just some other fluorescence change or very specifically FRET happening between the acceptor and the donor, i.e. we are actually getting the 2 coming together into close proximity. And this is an incredibly powerful tool for understanding protein-protein interactions, conformational change, and protein folding. So very big in the biophysics and biophysics space. So as an example, here's a kinetic evidence for a protein ligand interactions in the T cell receptor. So what we've got is the T cell receptor on top, and this binds to the major histocompatibility complexes with a specific peptide that's also bound and presented by it. And effectively, it's actually at the binding of this peptide with this complex here that is the binding that's going to be the binding event that's actually going to be studied here. What the researchers did was attach the donor on the MHC and the acceptor on the TCR, and then have the MHC and the peptide in one syringe versus the TCR in the second, mix them together and observe them coming together. Now, what we actually got was, what they actually got is these reaction right here. So what we've got on the right here is the fast reaction rate, the very fast time domain up to 0.2 of a second or 200 milliseconds of both the donor emission decreasing and the acceptor emission increasing. So clear evidence of FRET happening. Effectively, the donor and acceptor have come together. So that's indicating that very quickly, within this fast time domain, the donor and acceptor, effectively the 2 components, have come together and formed a transit intermediate. If you look at the longer time domain, there is actually a second process that happens on a slower domain that you can observe the donor emission, the slow phase over here. So what they actually end up with is 2 rate constants. One, which is the 2 components coming together at 30 per second, this to this, and then a slower phase going from here to here. Now that actually has some profound implications. Labeling the ligand and the protein allowed FRET-based monitoring of the binding rate constants. So obviously, a great way for looking at protein-protein interactions, which this really is. Fast kinetic-based analysis illustrated that there was actually a two-step binding. And actually, the first step is the majority of the FRET activity of the two coming together, and then there is another smaller change happening at a slower timescale. That second step is actually good evidence that there was a conformational change after the initial binding. So effectively, there is a biomolecular coming together of the two components, and then there is a separate, slower conformational change to form the final complex. Now, going to a completely different application, this is actually quite an intriguing way of approaching it. It was actually a study to understand the energetics and the driving force for the phospholipid bilayer versus simpler, purely fatty acid vesicles. So, is there just a natural advantage that drove the transition from primitive to modern membranes? Are they more stable? That kind of thing. And do vesicles spontaneously grow bigger when they have some phospholipid in them versus a vesicle with just simple fatty acid in it? So what did they do? Well, they took vesicles with a small amount of a labeled phospholipids, in particular a donor and acceptor pair on different phospholipids, and they were then mixed with unlabeled vesicles. Now, it'll come clearer in the next picture when I actually have the visuals for it, but basically, you've now got in a membrane two donor and acceptor pairs. Now, depending on the concentration in that membrane and the surface area of that membrane, you'll get different amounts of FRET as they come closer and further apart. And this can actually be used to calculate a surface area, and it actually can be given a standard curve by making up different vesicles with the labels in it with different concentrations of the donor receptor pair to get an idea of how FRET changes with effectively what they call a relative surface area, effectively how big the surface is and what the concentration of donor, etc., are within the membrane. So over here is basically the experiment. We've got unlabeled vesicle without phospholipids. The phospholipids are in yellow in this one, so it's the phospholipid containing membrane vesicle. So, and then you basically mix them together, the stop flow, and that's what that means. And then the phospholipid will get bigger, but the amount of label will stay the same, and therefore you're effectively diluting out the label within the membrane. And FRET should go down. But in this particular case, the surface area that's been calibrated will go up. And that's actually exactly what they see. So you've got over here is a vesicle containing a large amount of a phospholipid and also the label mixed with an equivalent sized, equivalent amount of an oleate only vesicle. And those controls where they only do put in an unlabeled but similar phospholipid membrane, and this black trace where there is nothing but buffer added in it as well. You can see there is no change in this blue and black trace. So what we're actually observing here is the phospholipid getting bigger with time. So it makes sense that if the phospholipid containing vesicles are able to effectively rob fatty acid from a smaller pure fatty acid membrane, then the opposite should be true. And that's exactly what happens. You're able to then take the same small amount of phospholipid, so it's not going to disturb the amount of, it's not going to be to the same equivalent as the phospholipid membrane, able to observe and mix it with unlabeled phospholipid containing membrane. Phospholipid membrane will get bigger, but the labeled non-phospholipid containing membrane will get smaller. And that's actually what we see here. There's the two controls on the top, buffer and with unlabeled non-phospholipid membrane up here, and then adding in the phospholipid containing membrane. Clearly, it's robbing the non-phospholipid membrane of fatty acids and effectively getting bigger spontaneously. So, phospholipid containing vesicles grow at the expense of non-phospholipid vesicles. Presence of small amounts of phospholipids may have presented a selective advantage to cells generally and actually support energetically bigger membranes. And really, it's probably the selective advantage of high phospholipid membranes over simpler non-phospholipid containing membranes. And it's basically the SX20 will be able to be used to measure the size of vesicles changing in real time. Right, the fluorescence. Now moving on to another technique, it's quite sophisticated end of things, the fluorescence polarization anisotropy to study RNA hydrolysis, actually unwinding. So a little primer on what FRET is. On fluorescence polarization / anisotropy. Effectively, if you excite a molecule, a fluorescence molecule with linear polarized light, you have selected a population of those 3 or 4 in a particular orientation. It doesn't immediately re-emit the light in the emission. There's a certain amount of time, usually called the fluorescence correlation time, where the molecule can move and tumble before the light is re-emitted for the fluorescent signal. In that time, if it's a really fast tumbling molecule, which the majority of simple fluorophores in solution, it'll get randomly, that selection will get randomly rearranged, and there won't be any bias in the direction of or the polarization of the emission. But if the fluorophore is slow in tumbling, or it's frozen, or any other thing that has, or in a very highly viscous solution, then that can result in a bias in the emission, in the polarization of the emission of the fluorescence. And actually, this is directly correlated with the tumbling rate, of the molecule. That tumbling rate has various things like a dependence on viscosity and temperature, which can be controlled quite successfully, which actually leaves the size of the fluorophore. So it actually is a proxy in a way of measuring differences in the size of the fluorophore. And because it's in stop flow and it's a fluorescence-based technique, we can actually do it in real time. The setup is over here, where we have 2 detectors. It's a T format fluorescence anisotropy polarization system. I keep saying anisotropy and polarization. Anisotropy tends to be the way of measuring, calculating a parameter that is the easiest to fit and actually deal with when analyzing the data. And that's the equation for anisotropy right there. But in the end, we've got the intensity of the vertical versus vertical and horizontal. Here, this G factor is effectively a term just to balance the detector responses between the 2 detectors here. So effectively, excited site, linear polarized light comes in to our cell block here, and then we were recording through 2 polarizers at right angles to each other. And that's the system. Now, here's a very interesting use of it. Now, what we've got is a dead box RNA helicase. Effectively, it's actually, this one is a specific one that binds to a hairpin called hairpin 92 as part of the E. coli genome, essentially. These dead box helicases are important in RNA molecule assembly, and in particular, this one is implicated in assembly of the E. coli ribosome, and effectively making sure that the correct RNA structure is formed and it's assembled properly. Now, how it does that is it binds very specifically to this hairpin here, and then using a bit of ATP, it unwinds, and that hence the helicase, the RNA. Now, what they did in this experiment to understand how it worked as effectively as an enzyme is create a substrate. In this particular case, it's got the hairpin, which is the recognition site. This is the hairpin here, and this is actually the helicase sitting in here in the structurally. But they added in a very clever way of measuring its activity in real time. So what we have here, is an 8-mer with a fluorescein label that is placed onto this helix, hairpin 92 and this helix 91 over here, that will be unwound effectively after this recognition on hairpin 92. So we label Hep92 with this small helix from the 8-mer, and it's equilibrated with the helicase in solution. So it's actually in syringe 1, but the enzyme reaction requires ATP to function. So you mix it against ATP in syringe 2 to initiate the helicase activity. And this unwinds the heparin and also releases the fluorescence 8-mer label. Because that is a double helix, but it will now be unwound. And that releases that 8-mer. And we're also going to have controls, no ATP in the syringe 2, and the 8-mer by itself with no other reagents. The important thing to understand here, of course, is when the 8-mer is released, its tumbling rate goes up significantly, in which its fluorescence anisotropy will go down, everything being even. And that's exactly what they observe. So here we've got just the complex by itself, but without the ATP, so it's effectively just sitting there, with no enzymatic activity. The 8-mer by itself has a much lower anisotropy, and it's over here, without the other parts of the complex. And then here's the complex with the ATP initiating the actual reaction. And here, this single phase drop in fluorescence anisotropy is specific to the release of the 8-mer. And basically, it can detect when that 8-mer is unwound by the RNA helicase and released from its other component. So what's interesting about this is that they've been able to use anisotropy to look at a very specific part of the reaction. There is other data that goes with this in this study that actually looks at other aspects of the activity. But by using anisotropy, they were able to very specifically say that that kinetic step is release and unwinding of the 8-mer. Now, obviously, there's other applications for this. Anything that you can observe with a big change in the size of the molecule, protein-protein interactions, nucleic acid cleavage or protein cleavage, and other interactions can be very powerfully understood with fluorescent polarization with some careful and clever experimental design. And that's basically there. So the conclusion is the SX stop flow spectrometer is a powerful tool to gain insights into complex mechanisms. It's a powerful tool for understanding chemical and biological processes generally. But I think one of the things that leverages its power is understanding and applying the right optical probe, specific labeling, or spectroscopic understanding of what you see, or what your fluorophore, or what your chromophore, what its absorbance should look like, and what the intermediate should look like spectroscopically, can give you a much more powerful understanding of the real process and eliminate or decide between different mechanisms for the process under study. And fundamentally, applied photophysics for the SX20 are leaders in the market for stop flow. And we have a stop flow solution to meet all your needs So thank you for your attention. And could I have any questions, please? Yeah, so somebody, very specific one, that's fair enough. How do you overcome the photo bleaching issue when using the SX20 LED? Turn down the light intensity. So basically, photo bleaching and fluorescence is an issue. It's a trade-off between the amount of light you can put in versus the stability of the fluorophore to that light. And photo bleaching is effectively degradation of the fluorophore. In the case of the LED of the SX20, there is actually a 20-fold difference in the light intensity you can set on it. So you are able to very simply turn it down. There is also options to put on filters and so forth if you really need to turn down the intensity even more. Obviously, the trade-off with sensitivity when you're doing that. So somebody has, if ligands bind to lipids on the cell membrane, can we analyze the ligand binding to the whole cell? I'd probably need to know more details specifically about what that mechanism is. But if your ligand was, say, a protein potentially, and there would be, or probably a protein doing some sort of cell interaction, there may be possibilities in using various labels to understand that. I just need to know a bit more about the system to get into the specifics. So how many maximum syringes can be used at once for the SX20 setup? So this is really referring to the double mixing, or sequential mixing as we term it. So the standard stop flow setup is a single mix system where you've got A and B syringes and a stop syringe, so 3 syringes. There is a possibility to add in 2 more syringes, and that actually is sequential mixing. What that allows you to do, so it's a total of 5 syringes in this case. One of them has buffer, and one of them, you have one of the syringes as buffer, one of them is a two of them are reagents that you're pre-mixing. What you can do there is, so you've got A, B, and effectively C. So A and B go together. They can be pre-incubated from everything from 50 milliseconds up to several minutes potentially, to create a intermediate state, or a very specific intermediate, and then that intermediate can be reacted again with a reagent C to understand it. By doing this, you can actually start to pick apart even more the reaction mechanism itself. As long as you're able, especially powerful when you have more than one reagent. So in enzymes with more than one substrate, you can add in one substrate, create an intermediate state, and then mix it again with the other substrate, which will then finish off the reaction cycle. And you can effectively understand different rates of the kinetics for all those components. What is the exact dead time for a 10 millimeter and 2? I don't know off the top of my head. I think as standard it's about 1.2 milliseconds. So what is the volume required for the cell? And obviously we have various types of cell. The standard one is a 20 microliter cell that has a 10 millimeter path length and a 2 millimeter path length, depending on which way you arrange the system. The dead volume in that one is actually only about 8 or 9 microliters, so I can tell you exactly the number. Can you perform anaerobic experiments? Yes. There is many ways you can approach that, or a couple of core ways. So a lot of people will actually set up the systems in anaerobic hoods in a completely anaerobic environment. And that has a lot of advantages in that it is very much a guaranteed anaerobic environment, especially useful if you're working with very sensitive materials. There is an intermediate way where you're effectively using anaerobic techniques where you effectively purge out all your solutions and loading syringes with nitrogen or argon, and load the system that way. You can also utilize the system without having to go to an anaerobic hood. There's more, it's probably not the best for the high sensitive materials, but if you're an enzymologist and you're working with oxidazes, it does allow you to have both an reduced protein, reduced enzyme, which you can then react with oxygen, which obviously you don't really want to do within an anaerobic hood. Can the SX20 be used in high pressure conditions? Unfortunately not. It's made to be used at standard atmospheric pressure and temperature. Obviously, you can vary the temperature over a wide range. How do you overcome issues with noise? That's a very specific question. I think depends on exactly what optical mode you're in. If you're in fluorescence or if you're in absorbance, what the source of the noise is. Generally, you obviously can acquire slower, but of course in a kinetic experiment you don't have that luxury. Really the only way is to put more light on the sample usually, and so you can either use brighter light sources, hence things like the LED, which may be right for the conditions you're after, or you can, in a monochromator setup, use wider slits, but there will be a trade-off with, some other aspect of the spectroscopy of it. Is there a super simple reaction you suggest for training purposes on the SX20? Actually, that nickel, for PDA experiments, that nickel triethylamine experiment is a good one to use. I think we do have some standard test chemistries that we can send you afterwards that are also very simple to set up and use and very easy to get a result from. So somebody's got a very specific question about, so we have a stop flow, the SF3 accessory for our Coruscant CD spectrometer, the V100. Is it possible to measure CD and fluorescence simultaneously? And the answer is yes. Now the caveat is you have to be using you have to be detecting the CD at the same wavelengths that you want to excite your fluorescence at. But yes, you can do that, or you can actually do it, you still have it set up with the fluorescence and the CD and just do two different experiments at different wavelengths where all you have to actually change is the excitation or the CD measurement wavelengths. Could the inner filter effect of the samples be a problem for fast kinetic fluorescence interactions? So this is the, so people who aren't familiar, the inner filter effect is effectively when you have a high absorbance and you're looking at a fluorescent signal, the absorbance will diminish the excitation light through the sample and actually skews the emission towards the start of the very geometrically to the entrance of the cell where the most sample is. And this can obviously affect the CD signal, the fluorescent signal. You're not getting a linear emission across the cell. But it can affect fast kinetics potentially, because you may get a change in the absorbance of the reagents, which actually is changing your fluorescent signal, but not in a way you'd expect. But most of the time, people are actually trying to use lower amounts of their reagents than the fluorophore, in which case it's usually not a problem. And the best way to solve that is to get your concentration to a point where the absorbance is not affecting the fluorescent signal. Does the software has the option to fit the spectra to various functions? Absolutely. So we've got two options here. You can, in the actual standard FX software, you can fit single traces to various standard reaction kind of mechanisms, 1 or 2 or 3 state functions. We also offer ProK4, which is able to take as a full spectra data from the PDA, for instance, or single wavelength data for that matter, but actually fit it to a model of your reaction process itself. So what you're basically doing, you're actually putting in A goes to B, B goes to C, C goes to D, and then it's able to take that model and fit that to the data itself. Can the instrument double as a standard fluorescence absorbance spectrometer for static experiments? It's something we've looked into. It potentially can. It's not necessarily the best way to use the instrument. Probably yeah, I don't think there's an option an option specifically able to do that. Okay, I think sorry, there's quite a long question here. I'm still trying to get my head around it. The substrate absorbs in the UV range and an intermediate product also absorbs in UV range. How do you tease apart absorbance from the substrate intermediate product from absorbance of the enzyme protein? Okay, so basically the issue here is really when you have multiple overlapping absorbances, how can you tell the difference and what's happening to each of them? I think what you're really coming down to is what you're actually looking at in stop flow is absorbance change. So you'll still observe a change in one of the components, even though you'll still have an underlying absorbance from, say, the protein itself. So as long as you can see the change, then you'll be able to measure the reaction coordinates. Now you could even get more sophisticated and fit the data, and you could actually then observe what or you can actually measure the protein separately and subtract that out from the data as well. Okay, so I think we're going to wrap that up now. It is 1:45. So thank you for all your questions, and thank you for your time. And as I said, any questions that we haven't got to, we will in the next day or so follow up with answers in an email directly back to you. Or if you have answers, feel free to contact us at info@photophysics.com. Thank you for your time.